Hey guys, in this video I'm going to be talking about how we generate plasma B cells, and the overall goal of this process is going to be discovering antibodies. And you might be asking yourself, why would I care about discovering antibodies? And one of the reasons you might care is because this is how hopefully in the future we will be able to cure cancer, or we'll be able to treat cancer and help your immune system identify tumors in much the same way we're able to use vaccines to help your immune system identify viruses before you're exposed to them. And so the very first thing that I want to get out of the way is defining cancer. And cancer, I have a picture of it here. It's kind of a little hard to see, but this blue region here is a tumor. You have cells, you have healthy human cells, and some mutation occurred in them for whatever reason, and now we have uncontrolled cell growth. Uncontrolled cell growth as well as division. And so that's the reason why you get tumors and they metastasize and they go all over your body and they end up killing people, because it is devastating. The veracity of life to grow and divide is incredible. But this is what's going on in cancer. And these cancer cells, as terrifying as they are, will hopefully have some features on their surface that your immune system can respond to in order to kill them before they get to the point that they end up killing you. And so this is part of the conflict that goes on all the time in you and everybody in life. And so for me, this is a really beautiful topic to talk about. Anyway, so we have uncontrolled cell growth and we have a tumor. And this blue region here is our tumor cells. And let's say someone had skin cancer, and what people will do in order to generate plasma B cells to start this process of developing antibodies, to discover antibodies that will bind onto the surface of your tumor so that your immune system can respond to and kill that tumor, is first take a biopsy of it. And this is kind of the future vision of where medicine is going. So we would take a biopsy of someone's tumor and we would inject these tumor cells into a mouse. And this is a current method we're using for research. In the future, hopefully we'll be able to do this with people, but there needs to be a lot of FDA stuff and further research to actually get to that point. But right now, we take a sample of our tumor cells and we administer a dosage generally into the tail of a mouse. And these mice here are bred in bubbles. And by that I mean they are grown in, they basically live their entire lives in very sterile environments. And the reason that you breed this mouse in a sterile environment is so that its adaptive immune system is essentially a blank canvas. And what do I mean by a blank canvas? Well, if I was to look at your immune system or anyone's immune system who's been outside within the past, however, since they were born, their adaptive immune system has been highly evolved. And evolved isn't necessarily a good word, but it's been highly exposed to a lot of pathogens out there. So you can have a lot of B cells and T cells and all these other specialized immune cells to recognize all kinds of pathogens that exist in this world. And as I said previously, there are incredible numbers of different types of pathogens out there. So your adaptive immune system is not a blank canvas. It is a very complex thing to look at. Now, if we grow a mouse in a sterile environment in which it's never been exposed to pathogens like bacteria or viruses, its adaptive immune system will consist mostly of naive B cells. And I do have a previous video on this, which I highly recommend watching before, if you're unfamiliar on this process. But these naive B cells are what makes this mouse's immune system such a beautiful blank canvas. And what will happen is that we'll essentially take our tumor cells and we will inject them into a mouse. And generally we'll have to do this multiple times to get the mouse's immune system to recognize that the tumor cells in the mouse are essentially a pathogen. And because this is the first pathogen, and hopefully the only pathogen that this mouse has ever come across in its whole life, then the adaptive immune system of the mouse is going to specialize in making cells to fight this specific cancer. And so this is a good thing, because now your mouse, after you've administered several doses of tumor cells to your mouse, will start creating plasma B cells that specialize in fighting this specific cancer. And so we will euthanize the mouse. Generally, this is done with carbon dioxide, and they'll do, they're going to open up the mouse afterwards, and what they really want are the mouse's B cells. And mice have trillions of cells or billions of cells inside of them, and there are about, we get out 200,000 B cells from this mouse, plasma B cells from this mouse. And so if I just blended up this mouse's spleen, and I know this is graphic, so I apologize if you're uncomfortable with this stuff, but if we were to look at our mouse's spleen and lymph nodes and maybe its bone marrow, if we were to take its spleen, we can pass it through something called a flow cytometer. And the flow cytometer, so we take its spleen, which will have millions of cells. So let's say this has like 5 million cells. Pass it into a flow cytometer, and these flow cytometers, which I prefer the word cell sorters, will take in millions and millions or billions of cells and be able to shine lasers on these cells that will help us identify what type of cell it is, so that we can pick out these 200,000 B cells in this haystack of 5 million cells from our mouse. And there are generally other steps involved, and these steps are called enrichment. And they're just ways that we can purify the plasma B cells in order to conduct downstream experiments to identify which of these plasma B cells are best at our overall goal here, which is killing our cancer. And so after we've killed our mouse, extracted its plasma B cells, enriched the plasma B cells, we can move on to actually doing what is called antibody discovery. And so antibody discovery is where we're going to, again, look at our tumor cells in another experiment and actually begin to see which one of our plasma B cells does a really good job of making antibodies that will have a high affinity for this tumor, so that we can identify those antibodies and then give those antibodies to a patient in order to actually achieve our goal here, which is to kill cancer. And this is a graphic process, but there's a lot of death involved in life. So this is a high-level overview of what is going on upstream in antibody discovery in order to help us identify plasma B cells. And once we have these plasma B cells, we can actually sequence them, figure out what kind of antibodies they're making that are so great at binding to our cancer, so that we can actually achieve our goal here and make cancer treatments and that kind of stuff. So I hope you guys find this useful. Thanks for watching, and let me know if you have any questions.